Okay, now that we're recording, I just want to show you all how you can get some graphics that you need from the Genome browser, for instance, for the book chapter that we're working on right now this summer. And just so everybody has consistently the same thing, because different people can get different gene information. So when you get to your Genome browser at Genome at UCSC.edu, it may be in different types of formats when you get there. Normally you get onto this landing page by going to that address. We are using this build of the Genome assembly, so I could put in something like frizzled 4. That's what I want there, the top one to look up. Let's go. Might get a table, or we get a gene right away this time. This is good. I want to simplify this. First thing you're going to do is turn off a lot of things like gen code. One good one to leave on here will be main in full. And I'm going to go and turn off some of the other things like conservation. What else do I have flipping up here? I'm just going through all the tracks and really just making sure I'm not really going to see anything other than I want. You can hit any of the refresh buttons to update. So right now I've just gotten myself down so I'm seeing the gene. I want to make things a little bit bigger, so in configure, I'm probably going to take the font size all the way to 24 and just show you what that looks like as a change. That's better. We might also go in here and do some other adjustments and configure. Right now I don't have any light blue vertical guidelines. You might. I'm going to make sure I don't. I'm not going to require a description above each track. I don't need to show the track controls under the main graphic. Although, I can actually, so we won't necessarily save that. So let's see what we got here. So I'm just simplifying right now the views that we have. And this looks pretty good. You can do another thing if you get a gene that will be many exons with large spaces in between them. For instance, NDP has lots of large introns and very tiny exons. You can actually, while you're here, hit on your keyboard, you can hit the letter E first, let go, and then hit E, and then hit V. I'm going to hit E and then V, and that's already put it in this format anyway. I might already be in this format. It will create basically this exon format for you. Yeah, it seems to be the same when I process both times. So if these had, if Frizzled 4 had a really gigantic intron and the exons look kind of tiny, it will respace a little bit of the introns and bring the exons into view for you, and it'll add different reference sequence numbers as well in the scale. I'm also going to do one more thing. I'm going to zoom out 1.5 times just so that you see you kind of have a little space around the gene. I'm going to process my EV again, make sure it looks good. So basically there's a good ideogram here of your chromosome, and there's the graphic of the gene. If this gene you were looking at on this chromosome, noticing the chevrons, if it wasn't arranged left to right and it was arranged right to left in direction because it's on the other strand, you could simply hit the reverse button and you get to flip your gene around accordingly. So we'll prefer from left to right, but now I'd like this picture and this chromosome picture, if possible, in a good file format that we can use by putting a manuscript together or putting figures together. And the best ones to get are go up to View, PDF, or PostScript. And what you can do is just use the PDF ones. You can get the current browser graphic in a PDF, and you can see there it is, and I can download that to my computer, which I've just done. You can also download the chromosome ideogram, which has your chromosome and has got a little red line showing you kind of the location of your gene. I can download that too. Those are the two little files that we want to have, and if we get those will make good diagrams in a gene reference section. And that's probably the best way to do that with the Genome browser. So you want things nice and big. So again, these are up at 24 font size, just so things are kind of large. When you get your gene, it will normally line up the beginning and the end of the gene in your view window. I like to back up 1.5 times, zoom out, just to give a little space around the gene. And let's see, that looked out pretty good for Frizzled 4. It should work out just about for anything we look up. LRP5. Here the EV method is kind of a little bit annoying. Here it's actually squished all the exons together, and that one might be more problematic to look at. And if I want to, let's see here, for viewing, I want to go, maybe I got it in help, the keyboard shortcuts. These are kind of handy. You can reset all your user settings. The exon view, I did the E to V. DV goes to the default view. I might go back to that. So let's hit a D and a V. That's back to the default view. So for something like LRP5, where the exons are all really small, but there's so many of them, I might just leave it without the exon view. And that works fine. I might just reverse this one here. So we have it that way. And I'm going to take off this gen code. There we go. So I just want the, under genes and gene predictions, the main version 1 is good. This is really just a reference gene. These are reference and ensemble manuscripts. They're just made to be kind of a nice reference gene with one drawn. But this would be a good figure to get for LRP5. And again, I got the chromosome. And something like NDP. Again, let me turn off all the things I don't need. Let's reverse this guy. NDP, you can see really the, zooming out a bit, the introns are much larger than the exons. Of course, this is a nice view to notice the thicker part of these exon bands represent the coding sequence. If you're not sure about that, you can turn on codes here for a second and see it's showing you where the coding is. So want everybody to realize that an entire exon is not necessarily used to encode a gene unless it's an exon somewhere in the middle of the sequence. I'm going to hide that again. But again, that's a nice view. If I use the E and then hit the V view on this, I think I have to have my cursor up here when I do it. EV, oh, maybe not. Let me go tap in here. EV. Okay, now it's not flipping on the EV function for me, but okay. EV. Oh, yeah, that worked that time. As you can see, it kind of shrunk one of the intron regions just for viewing, but I guess it's probably pretty good also in the default view. But here again, we've got our exons arranged from left to right, and that would be fine. Again, you can go to the view menu and get a PostScript download of each of these two files, and these can go in the text section where you write a little intro to the gene that you're working on.